Good morning, everyone. First of all, I really want to thanks everyone who was involved in organizing this award and selection procedure, and also organizing this whole presentation. I'm very happy and excited to talk about the work which we have done, which we pitched for this award, and and we got selected. So I'm very excited. So we'll talk about it in a minute. So first, I want to tell you a little bit about myself. I'm assistant professor. I have an engineering background in biomedical engineering. I used to work in a silicon IC fabrication before. Then I switched into applications in biomedical engineering and see how engineers can help to solve some of the problems in medicine. So that's the main idea, that how engineers can help to deal with the knowledge which is already out there and make something which is really practical and usable. So I have a lab there in Florida Atlantic University. That's my lab address. So if you really want to see some more stuff in other areas, so please feel free to go there. So before going into real technical details, I want to quote one saying from Muhammad Ali, that service to others is the rent you pay for your room here on earth. So that's really, really important. As a human being, we really have to put our energy to solve problems, help other people, and that's the kind of obligation of everyone. And as an engineer or scientist, from our side, we're also doing the same thing along with many other people's. I'll talk about, before, I just want to give you a little bit background about where was the need and why we are working on this project. If you look at this graph, it shows the prevalence of HIV all around the world. And the darker colors shows that they have a high prevalence, means more infected people with HIV. And then the lighter colors are like less affected. And one thing to notice here is that the mostly affected areas are the Africa region, and that region is also resource constrained. So it's a developing world, so they don't have resources and all that. If we briefly look at the numbers so far, right now there are about 37 million people still infected with HIV, and about 70% of that population is living in Africa. And about 54% of those people even doesn't know whether they have HIV or not, and they are affected. So it's a very, very big problem. We really have to have some diagnosis tests where we can quickly test those people so that everyone knows that what's their status. That's the one goal. The second is only 15 million people are getting drugs, which some of the drugs are available at the reduced prices by WHO programs, but you really have to know whether you're infected or not. And for that purpose, we need some quick diagnostic tests for HIV. So those are some of the backgrounds. One background slide where I just want to show you where is the need. And in this area, when we talk about it, we normally use very fancy and nice equipments in the developed world, like RT-qPCR, flow cytometer, and all that, to test for CD4 cells or HIV viral load, like the other infectious diseases. These systems works pretty well if you have a very well-established lab. But when we talk about the resource-constrained settings, all those countries where they don't have resources to do all this, then the idea comes to our mind, why we cannot have something where you can prick your finger, like a best example is a glucose meter, where you can prick your finger, you can quickly detect and quantify different chemicals in the blood. And but there's nothing out there as far as the HIV viral load is concerned. There are some of the tests available for antigen or antibodies, but for when someone is on treatment, we really have to monitor their HIV viral load. Because if HIV viral load is not falling down, we really have to switch the treatment, the ART, what drugs they are using, if their body is not responding well. So viral load is one of the most important biomarker for disease diagnosis and management. So the idea is why we cannot have these type of assays where you can use a cell phone, everyone has a cell phone, or some small portable sensor where you can quickly prick your finger, from a drop of blood you can detect HIV. And then we thought about papers. Like, why we cannot use the papers as a substrate, which is freely available all around the world, and it's very low cost, flexible. You don't have to worry about disposing them. You can just burn them off. So why we cannot use papers to build devices like a glucose meter to detect HIV and other diseases? So that was the idea. So these are the real chips, real devices which we have made. And the fabrication process, I will show you in a minute, is also very simple, that everyone, anywhere, can make these devices with very little infrastructure. So this is the idea which we worked on and we pitched for Humanity in Science Award, and we were selected as a winner, and I'll talk about it in more detail about the idea in a slide. So the basic idea behind how this device works is, this is the whole blood. So you have viruses, which are these bluish spherical balls, and then you have white blood cells, red blood cells, and a lot of other components in the blood. So we use a polyclonal antibody. We functionalize our magnetic beads with a polyclonal antibody. So this antibody targets P120. There is one specific antigen on the virus surface. So these specific antigens, it targets very specifically, and these magnetic beads, then you mix it with that, and after some time you can isolate those magnetic beads from a solution. And then for detection, for the rapid detection, we lyse those viruses with a detergent. So when you lyse the viruses, it will release ions, it will release proteins which are inside the virus structure, and then you can detect it with the impedance spectroscopy by using this device. That's the whole idea. So this is some impedance. Then when you have a control, or when you have viruses, you will get a different impedance at different frequencies, and you will be able to detect and quantify viral load. Okay, let me talk about first the chip fabrication. So this is the final design, where you have two electrodes. You have a 1 microfluidic channel. The channel thickness is about few micrometer thick. So we can take just one transparency paper, which normally used for a projector, and then you can make a mask with a laser cutter or with anything, because the electrodes are big, larger in size, so you don't have to worry making things small and nano or micro scale. Then with the spatula or conductive ink, it can be silver or graphene. With the spatula, you can just transfer this ink to the pattern, then you remove it and then seal it. And next slide, I'll show you the assembly of the device without electrodes, this image, but the electrode assembly is also very similar. So in this case, you have a one transparency paper at the bottom. On the top, you have another transparent paper. We have a DSA, which is double-sided adhesive, and you can just stick those paper together to make this microfluidic device, which is flexible. Then you can do many things with that. So it's so simple, very straightforward, low cost, and you'll see some of the results which are very interesting. So this is one video I want to show you. Okay, it shows that the channels are micron scale, so you will see a capillary reaction when we just put a small drop of blood. It will automatically fill in the channel. That's required. So it will fill up automatically. So you don't need any pump to do this job here. So it will automatically pull it inside. So this shows in the real time how these devices are flexible. So you can mix them also with the hand. So with the hand movement, you can mix the blood inside. So if it will increase the chances of capture of the antigen. If there is some cells inside, it will increase the chances. It will hit your target surface. So you are mixing it very well. So you don't need any mixer here also, because the chips are. If you have rigid chips, then you have to somehow vortex them or mix them. Okay, let's look at some of the results. With this technique, impedance sensing, we spiked different viruses. These are A, B, C, D, are different subtypes of HIV. They all are HIV viruses, but they are different subtypes. So they have a small genetic variations. And the good thing about this technique is we use a polyclonal antibody, so we can capture all different subtypes with the same magnetic beads. So when we lyse them inside the chip and we look at the whole frequency spectrum, and this is a phase, and you can see at certain frequencies, at the lower frequencies from 10^2 to 10^5, actually 10^4, you will see that the control, which doesn't have any viruses, and the ones which have viruses, different subtype of viruses, you will be able to see the difference here. And these are the results in terms of how much impedance magnitude you get. You will see in the case of a control, you have a impedance about 2 megaohms, and when we have viruses in the blood or PBS, then the impedance falls down or reduced, because once you capture viruses, you lyse them, your conductivity will increase, or in other words, your impedance will fall down. So that's why you will see a significant difference, and you can detect those viruses from a PBS sample. Then to show the specificity, whether this device works with all different viruses or not, because in the blood someone might have a hepatitis along with the HIV, so whether those will affect our results or not. So for preliminary results, we use Epstein-Barr virus as a control. We mix it with the HIV, and you notice that in the control, if you have a control, you have a high impedance. Then if you have an Epstein-Barr virus, you also have a high impedance. But when we put a HIV, that green line, you will see a significant difference. And when you mix HIV and EBV, you still see a significant difference than the control. That means our proposed technique, we can efficiently capture viruses we can separate viruses from if the blood has a different kind of viruses. We can specially capture HIV viruses in this case. Then in order to show that whether this technique works for whole blood or not, because it's really important, sometimes when we work in a lab, your project, you might have something working in a lab on a clean environment, like PBS or some solution which is not practical, but just to show the prototype. But in real sense, your device or technique should be working with the real clinical samples. And in this case, we will be using a serum or blood sample. So we took a whole blood, we spiked the viruses, and we were able to see that if we use a whole blood, we can see a significant difference between the whole blood control, which doesn't have any viruses, and the one which have HIV viruses. So that was a very significant results in terms of we don't have to use, in the case of a PCR, it's a long procedure, it takes a lot of time. The lab facilities, it requires a very high-tech lab, equipment is expensive, each test will take about $30, $50, which is a lot of money in developing world. And you need a technician to operate the qPCR or RT-PCR machine. So that's not really practical, and that's why 54% of people still doesn't know if they have HIV or not. So this type of a simple test based on the electrical impedance, where you isolate viruses quickly and then lyse them, and in a second you can measure their impedance, that would definitely change how the HIV is diagnosed or monitored nowadays. So I want to show you the current status. Right now we are testing this device with the clinical samples from Brigham and Women's Hospital, Harvard Medical School, through a collaborator. And because we have some patients there who are getting treatment, so we have to test this prototype with the real clinical samples from the patients who are on the drug treatment and see if the drug has any influence on it or not. And meanwhile, right now I have shown you some steps that first we have to isolate the magnetic beads and all that. In reality, we have to have something where you don't need those steps even. So for that, we are developing a cellulose paper-based, which is just a cellulose regular paper-based device, which doesn't involve the magnetic beads, but the paper itself, because it has a microporous structures, we will functionalize those structures with the polyclonal antibody, and then you just have to put a drop of blood on that, wash it one time, lyse it right there, and you'd be able to detect very similar as you do with the glucose meter. With the glucose meter, we just measure the DC current, we just measure the resistance. But in here, we are looking at the whole spectrum to see where we are getting the best results. Okay, that was one of the project, and we are trying to also improve the sensitivity. There are some other technical things which, if you are interested, I'll be more than happy to talk with you about, explain a little bit more about the details. But so this is the current status, and I briefly would like to talk about another project which involves the paper-based diagnosis, and which we have developed recently. So the whole idea is for that project is whether we can use those papers to develop some assays for bacterial and fungal infections. If you look at the number, how many people are infected, is a lot of 76 million people are infected with the foodborne diseases, and we normally don't detect them. Once someone has infection, then those people are treated. And once someone in a hospital, we have to use blood culture, gram staining, agar plate. So all that culture-based techniques, it takes about 3-4 days or even a week to make sure that someone is infected with what type of bacteria. So it takes a lot of time. Before that, we cannot give those people any antibiotics, or we'll give them broad spectrum antibiotics, which is not good. So we really have to detect those bacteria and viruses quickly. The culture-based methods are currently used, but they are not really, they are helpful, but we really have to speed up the process. And it also has some other applications, like in a peritoneal dialysis patients, people who have a kidney disease, they have to go through the hemodialysis, which is a blood cleaning, or peritoneal dialysis, where you use a peritoneal cavity with the cathedral, and you wash, you put some solution, and then your blood is filtered through this membrane, and then you can collect the waste. So in that way, you can also clean the blood. But the problem is, if someone has infection because of catheter, then you cannot detect it in this technique. How they do it now, normally right now, they look at the color of this waste. If it is cloudy, then we know, okay, someone has infection, so we really have to treat it. So, but it's very, very late when it's cloudy, because you can see it with the naked eye. So we really have to develop something where you can just put a drop of this waste fluid, and if you can detect if there is some bacteria or some fungus present in there, so you can start treating the patient way before then it's too late. So for that, the basic idea is that we use gold nanoparticles, and the gold is the property of the gold nanoparticle, how it interacts with the light, that if there are different sizes of particles, you will see a different color. So they all are gold particles. These are smaller, 5 nanometer, and then you increase the size. All are gold, but you see a different color. So that was the idea, that if we can somehow develop an assay where we can show the aggregation of the particles before and after, if there is a bacteria, if the bacteria presence show the aggregation, that will increase the effective size of the gold particles, and in that way you'd be able to see the color change. So for this one, for the gold nanoparticles, we functionalize with the LBP, which will bind with the LPS, liposaccharide, which is present on the binding site on the E-coli bacteria in this case. It can be another pathogen. Then you have to modify this antibody, and then we mix it, let's say, with any solution which has bacteria. And these gold nanoparticles, if there is a bacteria, they will go and bind with the bacteria, because bacteria has many sites for binding. In the long run, you will see aggregation of those bacteria all together. In other words, the gold nanoparticles come close to each other, and then you will see, let's say control is a pink, then when there is aggregation, you will see a bluish color. This is the control, this is a bluish color. Then you can tell whether there is a bacteria in there or not. So how this whole device works, this is normal cellulose paper. You first put the gold particles, functionalized gold particles, a drop of functional gold particle on the surface. So it will wet the surface. Now this paper is ready. Then you put a sample. It can be a serum sample. It can be peritoneal dialysis waste. Then you put that sample, and if you see the aggregation, the color will be different. Then we integrate it. Let's say from the color difference, you can tell whether there is infection or not, but you cannot really tell how many of those bacteria are present. For that case, we develop a cell phone-based small app which quantify the color, how much red, how much pink. And these are some of the results with the PBS, which is a very clean solution, with the Serum, which is a clinically relevant sample, and with the PD fluid. And you can see that as the number of bacteria are reducing, you will see a different 1 minus R square value. It means you have R square value 0.95. So this thing, 1 minus red valued by about 255 is a normalization. It kind of, you are quantifying the color intensity here. So as the number of bacteria are increasing, your color intensity is also increasing, so you're getting more bluish. So in that way, we showed that we were able to detect 10 bacteria per ml of a solution in just 15 minutes. So that was very, very interesting. Then now, the next goal is how we can, it's one bacteria, but most of the diseases like pneumonia and other diseases, they are caused because of multiple pathogens, not just one pathogen. So we really have to see 10, 15 different pathogens at a time to really make sure that this device is practical. For that purpose, we are working on it right now, that we can develop a little bit larger paper, which have one type of gold nanoparticle here, another type here, which will be already deposited. You don't have to really deposit it. If someone is selling it, the paper will all be prepared with the gold nanoparticles like a pregnancy test. So you already have a gold nanoparticles there. Once you put a urine there, you will see if there is a hormone present or not. So these particles will be already there on the paper. They will be stabilized by using a sugar solution, which we are also working on one of the project. We are using trehalose for that purpose. And in the end, once you put a sample, you would be able to see different regions where which one is, which pathogen is present, which is not present, whether we have E. coli, whether we have a staph, and all that. So that's the whole idea. So far, we have shown that it works pretty well for E. coli, but we really are now working on whether we can use this device for other type of pathogens or not. At the end, I want to just thank all of the people who are involved and who are helping and working on these projects. These are my lab members. Some of the graduate students, on the top, they are graduate students. So we have also some undergrad students who are also helping on some of these projects. We have also one very interesting high school student. He's also helping, a part of kind of more learning experience for him, and helping us on this project, on some of these projects. And I would also like to thank my collaborators. All this work was definitely a combined effort. A lot of people play their role at some stage. And so Dr. Maximum Caputi, he's a FAU, works with the HIV viruses, developing the cure. And then we have Dr. Hodores, he's in infertility clinic. Utkan Demirci, he's a Stanford, Hadi at Harvard collaborator, working on the similar projects. We have one guy, Jatin Vyas. He's working on the Candida or E. coli projects. He's at a Meshul General Hospital. And we have one Dr. Liu. He is at Lehigh University, helping us in doing some of the simulations. That was it. And thank you, everyone, and for coming here. If you have any questions, please feel free. I'll be more than happy to explain.